We have Amelia Marrero with us from Lakeland Regional, and she's going to talk a little bit today about the lab's journey and evaluation of the rapid AST platform from Q-linea called ASTar. So Amelia, take it away.
Hi, welcome everybody. I just want to thank you all for having me here at SCACM and Q-linea. As Ethan mentioned, my name is Amelia Marrero. I'm the microbiology supervisor for Lakeland Regional Health in Lakeland, Florida. So I did travel a little bit to be here. Excellent. All right, so today I'm going to be going over the technology and methodology behind the ASTar. I will say any questions that you have specifically about that will go to Tim and Ethan, but I will do my best to describe what's going on in the instrument. We started an evaluation in 2025 of the instrument, so I'll go over our data with that, our EA and CA and all that good stuff. I'll briefly go over some of the prior research that's been done about rapid susceptibility testing, including this instrument. And then impacts and future considerations are basically a study that we have conducted to see how this technology is going to help us at Lakeland Regional. And then last we'll do some Q&A. So just to kind of give you a little bit of background about Lakeland Regional, we are a quite large not-for-profit tertiary hospital. We have, it says 910 beds. I feel like we have more than that. We're quite big. We're smack dab in the center of Central Florida in Polk County. We are a level 2 trauma center, a level 3 NICU. We are a stroke center, chest pain accredited. We have a 32-bed physical rehab institute. The picture you see there is actually our pavilion for women and children that opened in about 2017, I want to say. We have 30 plus ambulatory locations, including a freestanding ED, with more to come. I know there's at least another 2 freestanding EDs that they're planning on building in the county. And then since about 2022, we are a GME facility. And then a little bit about our lab. So we are a full service 24/7 operation, as you can imagine. We have 5 pathologists on staff, histology, anatomic pathology, surgical pathology, our clinical lab, chemistry, immunology, including some special chemistry testing, hematology and flow cytometry, obviously urinalysis and coagulation. Our blood bank immunohematology is serviced by One Blood, which is a pretty big company in Florida. And then obviously our microbiology and molecular pathology, which is my home. So we do identification and susceptibility testing. We have the bioMérieux MALDI-TOF mass spec and Vitec 2 for susceptibility testing. We also do some confirmatory testing with Sensititer. We have rapid identification, serology, mycology, we're doing ID and susceptibility testing, and then microbacteriology, we do identify and then send out for susceptibilities as warranted. So we are a pretty big lab. You can kind of see their space is always an issue, as I'm sure many of you are dealing with in your laboratories. We also have a program with a local college, Florida Southern College, for MLS students. So we're kind of that ideal setting to bring in something like rapid AST. We're very, very busy. Our emergency room is the 2nd busiest in the nation. We just got beat out by like a hospital in Miami, but we were the busiest for a very, very long time. So, you know, obviously with so many patients, your physicians are wanting their answers a lot faster. So to go into the ASTar just a little bit, the technology methodology. So at a glance, fully automated touchscreen interface. You get rapid MIC results in approximately 6 hours. Again, if the instrument is full, it's going to take a little bit longer for the ones that were put on last. Very user-friendly interface. Hands-on time is less than 2 minutes, and I can honestly say that with confidence. It is very, very easy to set up and use. You know, quality control, maybe about 5 minutes, you know, just to get that set up. So how it works, so you've got your cartridge here. So your cartridge is where you're going to insert 1 milliliter of patient sample. So that's your positive blood culture, 1 milliliter right into the cartridge, which has buffers, dilution chambers, some Mueller-Hint broth on board. And then your next step is to add in a frozen insert, which kind of has a couple of different functions. It does contain supplements for fast-release organisms, although they're not doing any of that testing just yet. Enzymes and fluorescent dyes on that. And then you've got your disc, which is 336 culture chambers with the dried agents in twofold dilution. So you want to set up your positive blood cultures within 16 hours of it becoming positive. And then once you've loaded the cartridge and you froze it insert, you want to put that on the instrument within 30 minutes. So basically, that's it. It's all automated. You load your cartridge, you load your disc. The analyzer takes care of aliquoting the patient sample into the disc, and it undergoes some imaging, so my bright field microscopy, where it's going to determine the organism concentration. So once those images are captured, it's going to go through a very fancy analysis algorithm, which is going to tell you, what the drug, it's going to create your curves for your drug concentration, your blood drug concentration in each of the chambers. You can run up to 12 samples at a time, and kind of the great thing about it is that it's random access. So no having to worry and wait about batching and things like that. And with a 6-hour turnaround time that you're going to be providing for these physicians, I think they're going to be happy no matter how long they have to wait, for you to get these on there. So these are the current bug-drug combinations, and there are definitely more to come. And you can see it is pretty well stocked at the moment, and I'll kind of get into how we're able to get more drugs on there without necessarily needing to switch your reagent type. So we, like I said, we started this evaluation process in 2025, and we did do a side-by-side comparison. We looked at a couple of different instruments, and then we brought in 2 different ones to do that side-by-side comparison. And I will say off the bat that the EA and the CA compares very well to standard of care, and that was for all of the instruments that we looked at. And then we kind of went into the technical staff evaluation, which I'll get into, which is what helped to inform our decision. And then I'll talk a little bit about future prospects with the advanced user software. So if you know anything about Rapid AST, you've probably seen this information in some form or another on posters. They usually do a flow chart. But just our standard of care, the patient is drawn, for blood cultures. The blood bottle is incubated. We use the virtual bacterial alert system. Culture alarms positive. You do your gram stain. Then you're going to do your BioFire or whatever your Rapid ID methodology is. We do BioFire BCID2. We report those results to our pharmacy, our ID pharmacist, as well as to the patient caregiver, and they tend to get on board right away as far as recommending treatments based off of that result. And then you culture, which 18 to 24 hours later, when you have your growth, you can then confirm your identification with your biochemicals or your MALDI or whatever instrumentation you're using. So again, we do BioFire, Bioviri MALDI-TOF mass spec, and then we set our susceptibilities on the VITEC2. That takes overnight incubation. Then you get your susceptibility determination. From there, our process is to confirm any MDROs using the sensititer or e-test, depending on what the organism is and what resistance patterns it's showing. And then the patient's therapy can be optimized. With the ASTar instrumentation, patient is drawn, blood bottle is incubated, culture alarms positive, you're still going to do your gram stain, you're still going to set up your Rapid ID at that point, your whatever it might be. Then you set up your Rapid ASTar. So I know that it kind of looks like you can set up both at the same time. We waited for our BioFire because in order to set up Rapid AST, you want to make sure that it's monomicrobial. So if there's more than one organism growing in there, or obviously if you see it on your gram stain, there's more than one organism growing there, you're not going to want to set up the ASTar as well. But you could have 2 different gram-negative rods, which you didn't notice on your gram staining, and then you find out from your whatever your preliminary testing is that you've got more than one. Those are not going to be compatible with Rapid AST. But anyway, at that point, if you get your results within 6 or 7 hours, then the therapy can be optimized. So these were our overall results. These were the organisms that we were able to test, lots of E. coli, and mostly Enterobacteriales. We didn't have any positive Acinetobacters from blood cultures at that time. We did have some Pseudomonas aeruginosa. So total, we had 432 bug drug combinations, and you can see essential agreement and categorical agreement were excellent. We did have what we call an adjudicated EA and CA. So basically that was when if for whatever reason we had to run that patient on the sensitizer, we looked at that result because that manual MIC is going to be gold standard compared to the automated ones. So we were able to get it to go up just a tiny bit, but either way, we're well over 90%, which is what we were looking for. No very major errors, a few major errors and minor errors there. And then this kind of shows the breakdown of each of the drugs, how many organisms we tested against it. So you can kind of see with some of these fourth generation, some of the combination drugs, we are going to need to do some more. And so we're gonna pull out those AR bank isolates that are known to be resistant, that we have known values for, and do some additional testing so that we can validate the rest of those drugs. But I mean, again, we had some really great results, even with the smaller numbers that we had. So my technical staff loved this instrument. They appreciated that it was less hands-on time versus some of the other methods that we looked at. Very easy to process, as I mentioned. You're just taking your 1 milliliter and adding it to your cartridge and loading the cartridge in the disc. I really liked the storage. That was particularly easy. I don't want to go out and buy more refrigerators and freezers and things like that. So obviously the frozen insert, it's very small. The box that they come in can easily fit into whatever freezers you're currently using. No need for -70 or anything like that. Just a regular standard lab freezer works. And then the discs and cartridges are stored at room temperature. So very, very easy to store. What I really liked too, my educator heart really appreciated that my staff were empowered. Yes, I have day shift staff that read and report results every day, but my evening shift, my night shift, we're not currently doing anything like that. So they're very excited because they feel more like microbiologists, because mostly on those shifts they're doing things like serology and they're setting up, and maybe they're setting up AFB cultures, but they're not typically reading cultures, and they're definitely not resulting cultures. So this kind of excites them, this new technology, that they're going to be able to kind of interact more when it comes to patient care. And then we obviously went over all of this with our antibiotic subcommittee, infectious diseases, and went through kind of the stewardship part of it. So very quickly, there's lots of research out there on rapid AST in general, and in particular kind of started 2023, 2024, so I'm going to briefly go through some of this. But basically what we're finding out about rapid susceptibility testing in general, that you're getting consistent MIC results. It's helping to optimize patient treatment, helping with de-escalation, which is something that we really wanted to focus on. Mortality, morbidity, helping to lower that, and helping to lower length of stay. So what we already know, hospital mortality and length of stay associated with Enterobacteriales positive blood cultures. So this study was done by BD. It was published in Microbiology Spectrum in 2024. Basically, they evaluated the impact of how your regular standard of care, susceptibility turnaround time, and inadequate empiric antibacterial therapy in patients with bloodstream infections caused by Enterobacteriales species on in-hospital mortality and length of stay. So basically, certain variables that are within the hospital control can impact patient outcomes. Okay, we already kind of know that, but rapid AST kind of makes you look at this even more so, so where you can compare those times and see how much of a difference it makes. So inadequate empiric therapy, non-susceptible carbapenems, delayed AST turnaround time are associated with adverse hospital outcomes when the bloodstream infection is caused by Enterobacteriales. Another study that was published in JAMA, this had to do with early phenotype desirable antimicrobial therapy. So basically, is the timing of phenotype desirable antimicrobial therapy associated with clinical outcomes among patients hospitalized with Enterobacteriales bloodstream infections? So basically, once they knew early on that phenotype desirable therapy, or what the phenotype desirable therapy should be, these patients had way better clinical outcomes. So optimized care, we know, equals shorter length of stay, more favorable outcomes. So, and each day of broad spectrum therapy, we know as microbiologists that can lead to a higher likelihood of our multi-drug resistant organisms. And we want to avoid that. So knowing that, we know that we need fast and reliable and accurate susceptibility testing. So some of the things that Q-linea kind of wanted to show and what we are learning is that controlled inoculum actually has something to do with it as well. For carbapenem-resistant organisms, MIC values for meropenem and ertapenem were strongly affected by the testing inoculum. So the process that the Q-linea ASTar is able to do helps to make that inoculum more consistent among the wells, regardless of what the bioburden is in the blood, okay? Because we know that that is going to vary for your patients depending on what's growing and depending on actual patient factors. So this instrument allows a more fixed dilution, a more reliable dilution to give you more reliable MIC values. So now these are more focused on the ASTar, but basically it's several studies have been done that ASTar delivers on the promise of a faster turnaround time. So these were all posters that were shown at ASM, mostly. I think a couple of them were from MedID. But basically, the first one was a European study. So this one was versus the ISO standards for broth microdilution. But basically, we're showing reproducibility and accuracy with, again, your greater than 90% essential agreement and categorical agreement. The second study was Quest and Q-linea. This was a pilot study, so one of the first ones done with the ASTar instrument. But basically, the ESBL profiles of some of these more resistant drugs were matching very well with what the BCID2 gave them. And also they looked into hands-on time and runtime and were able to kind of, verify that much, much less than what you're doing with your standard of care. And then the last one was from George Washington University. And again, basically this was, proving that the instrument is reliable with your EA and your CA values, reliable for NDROs, and that you can accelerate your results by greater than 24 hours from standard of care. And again, a couple more studies. The first one was basically showing a categorical agreement of 95%. And then the second one just basically went into how reliable it is. Again, results from your standard of care compared to the results from the ASTar were across the board accurate. So now we can kind of focus a little bit more on impacts to patient care. We know that the ASTar is accurate. We know that you've got good agreement with your standard of care. So how is this faster turnaround time going to impact patient care? So the lifetime study is ongoing, but it's basically trying to determine on how clinicians act upon the ASTar results once they get these results much faster. So it noted an expedited workflow, actionable results in 31.4 hours faster than traditional broth microdilution with performance alignment, and it drove antibiotic change in 47% of cases. So you can kind of see there, 53% of the cases that they looked at, the ASTar confirmed prior therapy, which is good to know. And I'll show you some of our study data too, where that is something that is going to continue to drive patient care, is knowing that what you put them on mattered. What you put them on was the correct practice in the first place, the correct protocol. And that can drive how you treat patients, incoming patients, from that point on. And then antibiotic change in 47% of cases. And again, those are 47%, but it happened more than 24 hours sooner than it would have happened. So you can kind of see there what the differences were as far as antibiotic change, whether they needed to be switched, whether it had to be escalated, de-escalated, and then, whatever the dosage adjustments would be if they did keep them on the same drug. So just to kind of, this was done in Italy because there's not a whole lot of instruments here in the United States yet, but they did this study in Italy with 4 different facilities that had a high rate of antimicrobial resistance. So they wanted to ensure that, these were cases that it really, really does matter, whether they escalate, de-escalate, or make some other kind of change. So this was done against UCAS breakpoints. This was another study by Baylor Scott and White Research Institute. This was a 72 patient study, and then they also used the AR bank isolates as well. So this was compared against FDA breakpoints. So the potential clinical impacts, which is what we at Lakeland Regional are looking to focus on. But again, we know that it demonstrated performance agreement with standard of care, but it reduced the turnaround times, 13.3 hours versus 50.4 hours with standard of care. That's a lot of time. And they also found that it may lead to earlier de-escalation, escalation, or optimization of therapy and improve patient outcomes. So chart review, which is a retrospective review of what they had already done, suggests that earlier results could lead to exposure to fewer antibiotics, which we know will decrease their chances of developing an MDRO down the line, and then the associated adverse effects. So you can kind of list out there, there's a list there that what the outcomes that can be potentially impacted by the ASTar, and then potential clinical impact on the changes to therapy. Another one was done by Penn State Health, and again, the ASTar matches up with standard of care. This one they found that it was faster than standard of care methods by 42.2 hours, shorter time to de-escalation, and time to optimize therapy. All right, so based on all of what we had already seen, and after we had done our evaluation, to determine that yes, we like this instrument, we needed to go to our C-suite folks to, kind of explain to them why we need this instrumentation. So we did our own study, and basically we wanted to determine if the use of rapid phenotypic antimicrobial susceptibility testing will decrease the time to de-escalation in patients with non-multi-drug resistant gram-negative bloodstream infections. That was my goal, because again, from a stewardship standpoint, I want to make sure that these patients are not going to be on these broad spectrum antibiotics that are going to cause some issues down the line. However, I did this study with our ID pharmacist, who is phenomenal, and they wanted to know if we were optimizing treatment, because not every case is going to be a de-escalation case. In some of these patients, we need to make sure that they're on the correct therapy, or maybe they need to be escalated. So we ended up kind of roping those cases in as well. So we did a retrospective analysis. These were all of our positive blood cultures from 2024, lots of data to go through. But we kind of narrowed it down. So we did non-MDR enterobacteriales to determine the time to de-escalation or optimization. Because we knew, if they came off of the BCID with one of the resistance genes, it wasn't something we needed to look at. We know that they needed to remain on a broad spectrum or be on something even stronger. So the potential impacts of the study, optimization of therapy, de-escalation of empiric antimicrobial therapy, which may lead to fewer adverse patient outcomes. So obviously we also want to look at reduced costs and decreased length of stay. So we recorded everything. We looked at the time that the blood culture bottle was loaded, the time that it came up positive, the time, how long it took for us to gram stain, how long it took for the BCID results, what time the empiric therapy was started, and then every other step along the way to determine if and when a change was made to the therapy. So lots of information came out of this study. So not just, turnaround time and things like that, but really a lot of quality metrics for us. So you can kind of see up top our pathogen breakdown by percent. Obviously, E. coli was the highest, but quite a few other enterobacteriales. And then our results. So the graph on the left there shows that there were, out of the 363 total patients that we looked at, 214 were properly de-escalated. Okay, so that was about, I I can't see from here, 59%. And then 149, there was no de-escalation, okay? So then we further broke down the non-de-escalation into optimized, continued on narrow therapy, or missed opportunity. So optimized, our definition means that they were kept on a broad spectrum antibiotic due to their clinical status or patient-specific factors, or they were escalated to an antibiotic based on the pathogen isolated. For example, Enterobacter cloacae, cefepime is the drug of choice. So if cefepime were given initially for anything else, we would want them to de-escalate, not for Enterobacter cloacae. They're going to keep them on cefepime. And then CNT continued on narrow spectrum therapy based on BCID2 results, so negative for CTXM. So really, that kind of, we wanted to see that because we wanted to know what we're doing right from the get-go, okay? So if they got the BCID result and they have the patient on Rocephin, that's our drug of choice at Lakeland Regional. If they have the patient on Rocephin, they didn't see any other resistance factors, they're gonna keep them on Rocephin, which they should do, unless something else comes along later that tells them that they need to escalate. And most of the time, yes, BCID2 was negative. When we did finally get the MIC result from the VITEC, it matched up, they were good to go. We also looked at the patients that had a consult from our ID physicians. And you can see that really matters as well. So for those that were continued on narrow spectrum therapy, most of them were, with the purple bar, had an ID consult. Same thing with optimized and de-escalated. The ones that were properly handled in our eyes had an ID consult. The missed opportunities, they didn't. The majority of those did not have an ID consult. So this is something that we can take back to physician steering and say, hey, they're here for a reason. We need to consult with them more often. So, and with us bringing in rapid AST, we do think that's going to happen because all of a sudden they're going to have results so much faster. They're not going to know necessarily what to do with that. So this shows times now to either de-escalation or continuing on narrow spectrum therapy or a change in antibiotics. So these are median results. So for time to de-escalation, where again, T0 is our empiric start time of the broadest possible therapy, before they de-escalated was 48 hours, a median of 48 hours. That's kind of a long time. For those that were continued on a narrow therapy antibiotic, again, time zero was when we started that first dose of that antibiotic to the time that the standard of care VITEC 2 MIC report came out, because that basically just confirmed we were already on the right medication. So that's 57 hours. And then time to antibiotic change, this might be a little bit confusing, but again, T0 is an empiric therapy start time before some type of change was implemented. So we had actually 30 of those patients. 9 of them were de-escalated. Not 100% sure why we didn't move those to the de-escalation. I will have to ask my pharmacist why she calculated it that way, and I'm sure she has a good reason. But what I want to focus on is the 21 that were escalated. 16 had no change, but they were on optimal therapy, so we didn't include them in that time. And that was a 25-hour median time. So again, what we can determine when we look at this data, not using any rapid AST, is that for de-escalation, we can have results 30 hours sooner. For continuation on narrow spectrum therapy or to confirm that the first choice we had them on was correct, 39 hours sooner. And for optimization or patients needing a change, we can do that 7 hours sooner. So potential benefits, which we are going to determine once we go live with the instrument. So we'll look at another slice of data and make some comparisons. Decrease in adverse events that stem from the use of broad spectrum antibiotics, such as kidney injury, C. diff, antimicrobial resistance. Development of individualized treatment plans. We're really excited to kind of look at this on a case-by-case basis, just to see how we're all going to work together, including with our ID pharmacists, our ID docs, and the caregivers and lab, to determine how this is impacting patients on an individual basis. Reduction of costs, decreased length of stay, and decreased mortality and morbidity. So one of the things that we also did as a part of the study was to put together a business impact model. I'm not a numbers person, so I plugged and played, and Q-linea was excellent at assisting me with that information. And this is the stuff that you want to take to your C-suite. This is the stuff that you want to take to your lab directors, so that they kind of understand why you're pushing so hard to bring something like this in. So when I looked at that data, as an example, in 2027, we are anticipating 464 ASTar tests will be run in our lab. And that's a conservative estimate, because essentially we're just going live with Enterobacteriales, but we anticipate to be adding on Pseudomonas and Acinetobacter by the end of this year, but we'll also be doing testing on those as well. So again, there'll be more than the 464, but at that number, we can anticipate a savings of 232 bed days. And for each one, decreasing hands-on labor time by 2 hours. So, not going to go into numbers because that's going to be different for everybody. But with the length of stay, that's a fantastic thing to present, to the people that make decisions at your facility, that this could bring those down, because that seems to be the thing that they're always looking at, is how can we decrease the length of stay. There are also savings in materials and things that you're going to consider as well. But again, that's going to be dependent on how you use this technology. All right. So I just want to thank, these are the staff at Lakeland Regional that helped put this study together. My pharmacist is there, my partner in the molecular department, and my team lead. And obviously the scientists at the bench were the ones that really helped to inform this decision on what we brought in. And they've played ball the whole way. They're very excited about it. They love to see a new instrument being brought in, new technology for them to learn. So, and then obviously the team from Q-linea. It's been wonderful working with them. I've really enjoyed it. This has been one of the easier validations. As far as what we have to do, we're kind of always waiting on other people to sign off and build things in our LIS, and so that's always a little bit painful. But all of the support and everything that we've gotten from Q-linea has been excellent and really made a difference in our decision and bringing us on. Here are my references. And now we're open for questions. How fast did I talk? Really fast, apparently.